Before you can make a dream come true, you must first have one. Welcome to Lift Off MCN, podcast of the Trio McNair Scholars Program at the College of St. Scholastica. This is your host, Troy Apfelter. Today in episode 1, Research Foundations. 3, 2, 1. Now, I know that there are many new scholars out there who are currently gearing up for their first summer research experience, which is exciting. And I often hear new scholars talk about how excited they are about summer research. At the same time, these scholars will often come and express a general sense of what in the world am I getting into, and how in the world am I supposed to do this? And sometimes this latter feeling of fear and anxiety can be overwhelming and make it difficult to move forward on a project. All right, so to help with that, our goal today is to break down, demystify the process of choosing a research topic and appropriate research methods. And to do that, I'm going to walk across the hall to visit my colleague, Rachel Phelps Horton. And I tell you what, she totally wonks out about research. She loves talking about this stuff. She can go on, and it's amazing. And she loves to talk about students about how to do this. So let's go say hi. I am literally I'm walking across the hall. I have my iPad here with my earbuds in and my microphone connected. There's some folks walking down the hall. So this is an interesting look for me. And let's go check in on Rachel. All right. Hey, Rachel.
Hello.
How are you doing today?
I'm fabulous. How are you doing?
I'm having a good time.
Great.
Okay.
Take a seat.
Pull this over. This is totally like NPR. All right. So having a good morning so far?
Yes, it's very, very good. Thank you.
Very interesting morning, right?
It is.
So our new scholars are currently preparing for their first summer research experience, and they're probably feeling a little excited, but also maybe a little bit anxious. So you're an alumna of the Trio McNair Scholars Program at the University of Wisconsin-Superior, and you've done a number of summer research experiences as an undergraduate. So you've definitely been in their shoes. What was your experience like that first time when you were getting ready for your summer research project?
Yeah, great question. So I felt, first of all, very stressed out. As my first project, I really had my question and an idea of what I wanted to do, but nothing was really solid. It took a little bit of time for it to actually come together. So my first project was studying Duluth's architectural heritage and looking at different structures in order to nominate them for local landmark status for the city of Duluth. And so it was really interesting learning my own organizational process as well as learning actually how research independently is conducted. It was a great experience once I became a bit more comfortable with my idea and what I was doing and more comfortable with the freedom that was given to me. I was really able to develop that and work with my mentor, which really just solidified my love of research. And then I went on and did 2 more research projects after that the next following summer. So it was a great experience.
Nice. So you said it took a little bit of time to get comfortable in that space of being an undergraduate researcher. What led you to feeling more comfortable? How did you get to that point of feeling less anxious and more like, yeah, I can do this?
So for me, it was all internal. I suppose I had that idea where, I had to create the question that was going to astound society and, try to go above the level that I was at. And so I realized I was a bit more kind to myself, in that I realized you're not at the level of changing society with your ideas yet. You're still developing that. And so I was able to just kind of take myself out of what I was doing, look at the big picture, look at where I was at as a beginning researcher, understanding that this takes time, and that I'm doing great with what I'm doing. I mean, I'm already doing extracurricular activities, so that was in and of itself something amazing, and something I was successful or proud to be successful for. And so it was just kind of talking nice to myself, being kind and realizing this is my first time ever doing this. And it will get better and easier. I just have to take it step by step.
Yeah, totally. Be kind, rewind. That's what we used to say back in the 80s. Wonderful. So let's get into a little bit of the nuts and the bolts around research. And I was mentioning how you love to talk about this stuff. This is like your jam. So a good place to start is understanding the difference between primary and secondary research. So tell us a little bit about that.
Yeah, so primary and secondary research, they're very different, but they play together very similarly. Primary research is conducted around I suppose if you're in humanities, it's around the event that happened, as close to that event as possible, whether that is like a diary journal, a newspaper, an article, a video, well, depending on what time period it was, a letter. For the sciences, it's going to be more data set driven. So whatever that original data set is, will be your primary resource for your research. Otherwise, secondary resources, they describe, discuss, interpret, comment upon, analyze, evaluate the primary sources. And so a lot of researchers, beginning researchers, conduct a secondary literature review, understanding the field where they're at, what's been done, what's been said, and they use that secondary research in order to lay the foundations for them to build upon later the primary research that they will be hopefully in their PhD programs developing.
Yeah, that's great. And so a lot of the research that you've already done, you being the listening audience in high school or the beginning of college, is secondary research. You've done a research paper, gathered resources, analyzed that secondary research, and then if that's where you start as a summer research project, as Rachel was saying, that's a great place to start from, to then go into primary research, and sometimes students can start in primary research. So it's kind of where your project takes you. Excellent. Now, if you start hanging around researchers a lot, which Rachel and I hang out with faculty a lot, and you'll start to hear them talking about quantitative methods, qualitative methods, mixed methods, and who knows what else methods. Some $5 GRE words here. What's that all about, Rachel?
Great. So yeah, big words. When you get into the research field, a lot of new vocabulary will become very apparent to you very quickly. And so I'd like to discuss qualitative research real quick. So that is an approach for exploring and understanding the meaning individuals or groups ascribe to a social or human problem. So this process involves emerging questions and procedures. The data is typically collected in a participant setting, and the final written report has a flexible structure. So different kinds of qualitative methods would be things like interviews, ethnographies, surveys being done. And depending on which approach you take, it can very well, be divergent in that way. In looking at quantitative research, that would be an approach for testing objective theories by examining the relationship among variables. These variables can be measured typically on instruments so that numbered data can be analyzed using statistical procedures. So when people think of quantitative data, they think a lot about numbers, data sets, variables, a lot of psychology, sociology, neuroscience, a lot of the pure sciences will be working with quantitative research. And such, there are various methods depending on exactly what quantitative design you're using, such as experimental and non-experimental designs, and other things.
Yeah. So there's a lot to this. Don't get overwhelmed. Again, talking to your faculty mentor is going to be the best way to learn this and to just get in and start doing it. So you'll get there. But right now today we're just trying to introduce this to you, so you hear the word quantitative and you're like, oh yeah, I've heard that word before. Something to do with numbers. Good. You got that down. You're solid. All right. Well, for the rest of this podcast, we're going to check in with the faculty panel that we recently hosted here on research methods at St. Scholastica. So, Rachel, can you tell us who was in the room, which faculty members were in the room at the panel that we're going to now listen to?
Yes, of course. So we had Dr. Karen Peterson from psychology, Dr. Daniel Westholm from biology/biochemistry, and then Dr. Jennifer Mackey, who is in chemistry.
Cool. All right, so let's get to it. Thanks, Rachel.
You're welcome. Enjoy.
Right on. To get started, we asked the faculty panel to give an overview of their research career and trajectory to this point, starting with undergrad and going through their current position in academia as a faculty member. And Dr. Karen Peterson got us started off.
Well, I feel like I should have brought my CV in with me today. So, let's see. Before I went to graduate school, I did research in a lab that was looking primarily at stress and pain. So trying to figure out the relationship between stress and pain and looking at gender, and it was experimental. So we actually induced pain in the research subjects. People volunteer for that, if you can imagine. Using a few different methods. An ice bucket works really well. We also did some electrical stimulation to cause pain. I know it sounds really evil. And so that was fun. And then I moved from that to graduate work where my work focused on cardiovascular behavioral medicine, so understanding psychosocial risk factors that contribute to and predict cardiovascular disease. And I did a lot of work actually with U.S. Census data, looking at qualities in people's neighborhoods and how that related to biomarkers for cardiovascular disease. And so we had blood samples and went back to neighborhood and individual level data. That was a huge data set. I think we had 2,000 people. So that was really nice. And I've kind of bounced around, honestly, since being at Scholastica and not having really the same resources that I had at other places, and that's pretty typical of what we do. So I've done work on mindfulness training and health behaviors, so whether or not how mindful you are affects how much you sleep, exercise, healthy foods that you eat, things like that. I've done some work on probiotics and whether or not ingesting probiotics affects your stress response in a laboratory situation. That's the most recent study that we did, and we gave some students placebo, some students real probiotics, and looked at outcomes that way. That was just published, which was a good finding. So I've kind of bounced around. Those are a few things that I've done. I know it's hard to summarize it all.
Yeah, I've kind of been all over the place with my interests and a lot of my research experiences. I always knew I wanted to be a biologist, but as an undergrad, it was difficult for me to figure out which direction I wanted to go. Probably would have been best served maybe just sticking with biology, but as at an institution where you could major in different biological specialties, so it was interested in ecology, I was also interested in microbiology. And I had a lot of tension and difficulty deciding between the two. Ended up majoring in microbiology, but I was lucky enough to have 2 research experiences as an undergrad in ecology, looking at population genetics, and then in a virology lab of my undergraduate advisor. And so I really liked those experiences. I knew I wanted to stay in a research setting, but I wasn't quite sure, even after I graduated, which area I wanted to enter graduate work in. So I worked as a lab technician for 2 years, researching transport processes at the blood-brain barrier, and I ended up actually staying in that lab that I worked as a technician during grad school. So in grad school, I was more of a biochemist, so I went from ecology, microbiology to maybe more biochemistry. I was looking at what are the certain structural motifs important for recognition by a transporter that brings things in and out of the brain. And then as I was wrapping that up, a job opened up here in microbiology, and so kind of went back to my roots, and I've been back into the field of microbiology. So today, most of my research interests are related to bacteriophage biology. So these are viruses that infect bacteria. And I do research both in the classroom setting, but also with students that have finished my bacteriophage courses, that have started research with me in the classroom and then have become trained and are ready to move on to those next steps. So the primary topic of my phage research is basically comparative genomics amongst a variety of different bacteriophages that infect a certain family of bacterial hosts. So this project is kind of like a crowdsourcing project. I'm involved with students here on campus that are collaborating with students at about 100 different institutions that are all doing similar sorts of research. So instead of being in these isolated kind of silos, we're combining all of our data and really increasing the power of our findings. So far, this is called the C-Phages program. Our institution, along with all these others, we've identified thousands of new phages that have never been described before, and about 1,500 have been sequenced and deposited on the GenBank database. So it's an interesting way of approaching research, highly collaborative and cooperative too. We aren't competing with each other, which is sometimes you get that idea that applying for grants and then doing research, you're competing with everyone else. That's really not the case at all with this model.
Right on. So my research experience, I would say, started out in undergrad. I was studying for a Bachelor's of Science in Chemistry, and I did a biochemistry emphasis. They didn't have it as a degree, so that was kind of an interesting thing that has changed in my lifetime, that's become offered. And I've always been kind of an interdisciplinary kind of person, and that was certainly coming through as a kid too. I like to do a lot of different research things, and so I did a little bit of tutoring in like the math lab, which is like a writing lab for math classes, and then I did a number of different projects as an undergrad at UW Superior. I studied enzyme immobilization on, it was kind of like an industrial application. If you could immobilize an enzyme on a surface, you could pass stuff over it and basically catalyze a reaction. So that was one of my projects. Another project was inorganic chemistry based, and it was like very moisture and air sensitive, and so I had to work on a Schlenk line, which is completely keeps all of the air and water out of your reaction. That was exciting. I only started one fire. And then I worked on kind of a microbial project where I was looking at how well certain microbes could digest diesel fuel. That was another project of mine. And then I think that may have been the 3. That's a lot for entering the undergrad anyway. But I also tried a number of kind of internships. I got an internship at water and soil testing facility, and so that was like my part-time job for a while, gathering samples, going and picking them up, actually, like driving around town, and then bringing them back and analyzing them in the lab. And I also worked at Chromoline, which is now called Iconics. They do light-sensitive emulsions, and so essentially paint, but light-sensitive paint. It's very technical and has a lot of chemistry involved. And so the man who's in charge of the lab there was a PhD, probably organic chemist, and so that was kind of my boss, although I was a couple of people removed. So that was a fun exposure to industry, although it really helped me decide that I didn't want to go into industry and that I wanted to go into academia. And so all of those, I would encourage you to get as many experiences as possible of diverse, having a diverse background in terms of, am I at a federal agency, or am I at the academic institution, or am I in the private industry? Those can have much different flavors. Through all of that, I was most excited about biochemistry, but to be honest, the way that I was making decisions at 18 was I was watching my older brother go through biology, no offense, Dan, and he and all of his friends were getting jobs with their chemistry minors, and so I majored in chemistry because I was like, I'll just cut to the chase here and get the major that I can just go get a job right away. I wasn't sure I wanted to go to graduate school, and so but I did, and my reasoning on that, going immediately instead of taking a gap year and working in a lab or doing a job, which probably would have been good for me, my reasoning was I thought if I got a job with a chemistry bachelor's degree, I would make decent money. I'd probably make what I make now, and I would never go back to school because it would be too comfortable. So I went straight to graduate school. I went to Iowa State University, and I worked in E. coli, and so bacterial model system is really handy because your bacteria double every 20 minutes, and so you can start a culture at night, and the next morning it's ready to go and be used in your experiments. So I found that really useful, obviously, because you could have multiple generations, as opposed to people like, since I was at Iowa State, there were a number of people who worked on like corn as their model organism. They would get 2 crops a year, 1 crop in Iowa and 1 crop in Hawaii. So the good side was they got to go to Hawaii every single year. The bad side was they had like 2 experimental setups a year, and that would drag out the program. So what I studied in bacteria was actually ribosome assembly. If you're familiar with the ribosome, it's the machinery in the cell that makes proteins. And so it's really not still even now well understood how it assembles. So I was working on, actually, the ribosome has 2 pieces. I was only working on 1 piece, the small subunit of the ribosome in E. coli. And it also intersected with some chaperone protein work. So I used a lot of techniques of biochemistry and molecular biology, and a little bit of flirting with genetics as well. And so I've always been interested in the places where different disciplines intersect, and that was certainly a good program for that. A lot of the programs that I see our biochemistry students go into are the graduate programs that they all have a bunch of letters like BBMB or BMB, and they usually stand for biochemistry, molecular biology, and sometimes they'll have biophysics in there too. And so that was the case with me. And after that, my first job, I went and taught for a year at a community college and found out that I really wanted to still do research, so that wasn't a good fit. And then I got the job here at Scholastica, and I've been here ever since, which is like 14 years. I've done a lot of bouncing around in my research. I think Karin was talking about that, and so that's definitely true of me. I started out with an offshoot of my graduate research and found out very quickly that it was too expensive and also too competitive. And so I couldn't keep up with that cutting edge kind of research, and I had to reinvent myself. Some of the best things I've done have been collaborative and interdisciplinary, so there's a real theme. I worked at UMD one summer with Lou Barker. She worked on Mycobacterium marinum, which is a model organism she was using to study, gosh, I can't even remember, what is the infection that you would use Mycobacterium for?
Well, there's TB or...
Yeah, TB. So tuberculosis, that was a long time ago. And that was cool because I got to do a lot of work with biofilms, which I had never used before. And biofilms, essentially, like if you think about a rock, if you pick a rock up out of the lake and there's slime under it, there's a bunch of bacteria that lives in that slime, and the slime is a biofilm. That's kind of their environment to live in. And those can be really tricky in terms of infection in the human body too. So biofilms, knowing how they work and how you can disrupt them is really important. So that was kind of a more medically based research project. I also collaborated with Clay Carter at UMD, and he was a buddy of mine from Iowa State, and so he worked on plants and the model organism Arabidopsis, which is a weed. You usually have model organisms that reproduce easily and quickly, and so weeds are good for plant studies. So I did a little bit of work with him and eventually got a paper out of that, which was really pretty sweet. I brought a student from Scholastica with me, and we worked side by side full-time in the lab, which was a really neat experience. That was under another NSF granting kind of thing called the Research Opportunity Award, which is a really neat mechanism to get funding from. And then last year, I went to the Environmental Protection Agency and spent my sabbatical research year there. And I worked on CRISPR-Cas9. Have you heard of this? Any of the biochem people? Should look up CRISPR-Cas9. It's a new as of 2015, it was the science breakthrough of the year according to Science Magazine, so quite new. And it is a way to mutate genes in pretty much any organism you want to. And so I worked out in my year at the EPA, I worked out a method to do in vitro fertilization for fathead minnows and genetic mutation of fathead minnows using this microinjection system of this CRISPR-Cas9 enzyme that can go in and basically cut the organism's DNA and cause mistakes in a gene that you're interested in targeting very specifically. And they work on the fathead minnow because it's a regionally relevant organism for toxicology studies, because it's something that can live out in the Great Lakes. And so that's why, like, normally people would work on zebrafish, and zebrafish is not native to this region, and so they work on fathead minnow. But not as many people work on fathead, so it was a whole new thing to be like, can we do IVF? Can we do CRISPR-Cas9? The neat thing about that whole project was, number one, it worked, which nothing worked for 9 months. So out of 12 months, I knew I was like, okay. I mean, I was making obviously progress, but I made the breakthrough, it's going to work at the 9-month mark. But it enabled me now to bring that back to Scholastica, and I've incorporated it in the biochemistry curriculum. And the reason that I find that important is it's very much like the phage class that Dan is doing, where I get to organize and direct the research of 30 individuals instead of 1 to 4 that I used to be able to handle in my own little research lab. And so it gives them good training. In the first semester, they get a lot of the bioinformatics training that they need to figure out how to design the tools that we essentially inject into the fish. And then the second semester, the fish work is all still happening at the EPA, so then we get basically fish from them, little tiny frozen, 14-day-old fish. They fit in little tubes this big, and they're at the very bottom, and you can barely see them. We extract the DNA and then amplify the target that we were trying to hit and do the analysis to determine if the sequence has been altered in that target. And so after all of that, very similar to Dan, I then can recruit people who have had those 2 semesters of that project, and then they can work with me in the lab. So I have 2 people for the summer in the summer research program, and in the fall I have at least 1 person registered to continue working in the fall. And so they'll help me to continually modify the curriculum as well as pursue the research. And the EPA is continually collaborating with us, and we're bringing students over there and visiting and seeing the setup and learning about all of the science and what it's like to be in a federal government facility research lab, which is something that was totally new to me last year. So.
Cool. So after Dr. Peterson, we heard from Dr. Daniel Westholm and Dr. Jen Mackey. And what I found interesting is that in all their stories of their research trajectories, you see so much variety, so much changes of course, depending on where they are in their research careers, resources available, what they're trying to do. And so I think that's really interesting for our new scholars to keep in mind as they're preparing for their first summer research experience. You're going into this and you do not need to solve all the world's problems in the first 10 weeks. This is the beginning of a long journey of discovery, and it's all about a process. From there, we went on and we asked the faculty members to expand a little bit on the variety of research methods within different subfields within their discipline.
Sure. I can probably speak best to my own discipline. And it's interesting in our department, most of the research that is done is kind of more biological or cognitive, which isn't necessarily true, but there is social psychology, which is I honestly know very little about. So Deb Schrader and Lori Anderson teach social psychology here. Has anyone taken it yet? It's interesting. It's all about these social factors that you wouldn't hypothesize or expect to affect behavior, but they do in all of these very predictable ways. And that includes things like stereotyping is a big area of social psychology. And a lot of interesting research is done in that. We are doing a faculty search right now and had a number of social psychologists apply for the job, and we're not looking for a social psychologist. So, but got to read some blurbs on the research that they're doing. I think it's fascinating. It can also get into the realm of political psychology as well, which is obviously especially interesting these days. Cognitive psychology, I think, is slowly dying a little bit and merging with cognitive neuroscience. So we used to do all these fancy pure cognitive psych experiments trying to understand how the brain influences behavior. And now with the methods available in neuroscience and imaging, various imaging mechanisms, and being able to really see in real time how the brain, what part of the brain is more or less active, depending on how a person is feeling or what experience they're having, what behavior they're engaged in. There's less of the kind of fancy methodologies associated with cognitive psych and more going on in neuroscience. I really am a biological psychologist and a health psychologist and a clinical psychologist. So my degree was in clinical and in biological and health psychology. So I worked with patients all through graduate school and for a little while after, treating depression and anxiety and schizophrenia and all of those things. But I also, my research was always focused in biological and health psych and understanding how that sort of the interplay between health and disease and our brain and behavior, broadly speaking. I'm trying to think of what areas of psychology. There's personality psychology, trying to understand more stable characteristics. What else is there?
Ed psych? I don't know if that...
So that is educational psychology is really far outside my realm of expertise and is typically a different degree. It's typically a doctorate in educational psychology, not a PhD. We have students go on to grad programs in educational psychology, working in the schools, testing kids with trying to figure out what learning disabilities they have or special learning needs they might have. It's an interesting area. Those are, I think, the broad. I know I'm missing some things, but
It gives a sense of
there's a lot
the
out there.
and the breadth of the field.
Yeah.
And how about biology, Dan? You touched a little bit, talked about microbiology and some of your time dabbling, or not dabbling, but biochemistry. But
Yeah, it's a really diverse field. I mean, you're basically working with anything from molecules to ecosystems.
Mm-hmm.
So huge scale of study. More and more there's been collaboration between these various fields. Like as an undergrad, I felt like I was either going to be a microbiologist or an ecologist, but I mean, there's microbial ecology, or we're looking at ways that microbes impact nutrient cycling and all sorts of things. But the traditional disciplines, I would say, would be defined as biochemistry. That's kind of a little bit hazy. Here it's housed in the chemistry department. Other places maybe it's housed in the biology department. But moving from there, you have molecular biology, which is basically looking at how are genes expressed, looking at DNA and RNA and how that's turned into proteins. Genetics, which can incorporate aspects of biochemistry and molecular biology. There's classical genetics, looking at how inheritance works and that sort of thing, and traits are expressed, but also molecular genetics, looking at the molecular basis for all those sorts of things. You have microbiology, which is the study of forms of life that are too small to be seen with the naked eye, so usually bacteria, viruses, fungi, sometimes algae. Those are the primary forms of life studied under that banner. And then moving beyond that, you get into ecology, evolution, and behavior sorts of disciplines. So this can run anything from animal behavior, looking at what are the various types of animal behavior, how are those things determined, how does that impact their life history. And also ecology, looking at anything from community ecology, how different populations interact with each other, to nutrient cycling in the ecosystem on a global scale. And then there's fields of evolution. Again, that can take a molecular approach, and that's kind of what I do to a certain extent in the phage class. I look at how do these phage genomes evolve, how do they change as we look across different hosts that these phages infect. Remember, phages are viruses that infect bacteria. So we can look at how genomes have evolved as host preferences have changed, or as we look at different parts of the globe. But we can also look at evolution from a much longer time scale, looking at these macroevolutionary changes where whole new species are appearing and disappearing. So it's a huge field. The nice thing about biology is you don't, as I'm evidence of, you don't have to specialize and stick with that specialty your entire life. So as an undergraduate, if you would have said I was going to be a biochemistry graduate student, I might not have thought you were telling the truth, but that transition occurred pretty easily. And then as a graduate student, if you would have told me that I was going to be teaching microbiology, I would have said I would have really liked to do that, but it wasn't necessarily something that was first and foremost in what I thought where I'd end up, especially the field. It's kind of one of my first loves in biology, but so there's that aspect. But there's so many opportunities to change direction, to change training experiences. So, and that's true in lots of fields, but in biology, that it definitely holds true as well. And I'm kind of veering all over the place, but with something Troy said about as an undergraduate, when you're selecting your research topic, or you're just falling into a research topic, so a lot of times you aren't necessarily selecting what you're doing. You aren't designing things from the ground up. It's sometimes intimidating figuring out how. I know as an undergraduate, I always wonder how in the world do people end up studying this tiny little thing? How do they think of that? It seemed impossible for me to, like ribosomal assembly. Like, how do you focus on that one little piece? And it slowly, you're rarely is anyone synthesizing all these ideas by themselves. You're taking your experiences, what other people have done, and kind of building upon that. So you don't need to feel like you need to, if you aren't at the stage in your development where you can think of these research topics at that level of specificity, don't panic. That slowly, it just slowly kind of develops and builds, and then one day you'll think, all right, I have an idea, and I'd like to do this. And it kind of happens organically. Like, you don't need to. I don't think you need to worry too much about that at this point. I know I kind of did as an undergrad, and it just was one of the things that made me kind of think twice about entering research. Like, can I think of these questions that are that unique that no one else has thought of before? But it develops with time and practice.
So, long story short, you may fashion yourself a biology major, but what you're going to do research in now and in the future is nearly infinite possibility. All right, so then as we moved into the latter part of the faculty panel, we asked the faculty to talk about ideas or strategies for connecting with the faculty mentor and getting that first project up and going. So to get us started, we have Dr. Mackey.
I think my experience was just joining someone's lab, and they have projects that they're doing. Mm-hmm. And so usually they're like, you may do A, B, or C. Mm-hmm. And then you choose which project sounds cool, and you just go with it. And as an undergrad, you're going to have a ton of supervision, and that's totally appropriate. And your job is to ask a million questions. So whenever something doesn't make sense, you ask a question. And you have to ask I don't know how many million questions before you get to the next level, you know? And I still feel like I'm not always designing what I'm going to study. Like, I collaborate with people, and so they know what they want to do, and I know what my skill set is, and then we match it up, and it works. So you don't have to really worry about it. The only thing I worry about with my students in the lab is if they feel scared to ask a question. That's when when time gets wasted and things go wrong and materials get just used up, and we have to buy more and we don't get anywhere. And that can happen in the normal course of research, so you just really want to be open. If you don't understand something, advisors are there to advise and to explain things, and they want you to learn. And so I just felt like that was part of my development, that I was just like, whoa, this is a stupid question. It's not. There is no stupid question, and everybody's been at that different place in their development and can relate to it. And no one's going to judge that unless you don't ask the question. Then they might get mad later if there's a pattern of not asking the question, which I could have seen myself do as a student, which is why I say it.
No, I totally agree with those comments. As I mentioned, I had two experiences as an undergrad. They were very different in the sorts of things that we were doing, and, you know, the research topics and the techniques, and the sorts of supervision we had. So I agree, as an undergrad, supervision is key. In one of those experiences, though, I didn't have as much supervision as I think most undergraduates would. In some respects, it was kind of liberating, like, oh, I'm kind of in charge of what I'm doing. But I always had this question, why am I doing this? Half the time I did not know why. I mean, I knew what to do, like the various things to put in the wells, but I had no idea why I was doing it. And so if you have those questions, if you have those moments, definitely ask the question. And part of the problem with that particular experience is we had a graduate student that was supposed to be supervising us, and then she switched labs, so we were just kind of like left. And then the other experience, it was very it was a really nurturing environment, well, lots of oversight. So both the principal investigator that I had and then the graduate student really took an effort to make sure that I understood the questions, why we were doing these things, the big picture sorts of things before we even started the actual day-to-day lab work. So those are key questions to have answered right at the start, because it makes the research experience so much more fulfilling if you have answers to those sorts of things right from the get-go.
Yeah, and I think if you do have the luxury of designing your own project, or the challenge of designing your own project, the advice I got, which is the same advice that I give, right? We repeat what we learn, is really just echoing that sentiment of start small. That's what I encounter every time I work with students designing projects, is they just want to go big, they just want to answer it. And your first couple of projects are really about understanding the process and from start to finish, and it's most likely not going to be super novel at that point or super exciting, but it will be exciting to you because the first time you run your analyses and you get results, there's nothing like that moment, it should be fun and exciting and challenging, but not too overwhelming. So asking questions and keeping it small and manageable is, I think, the key to success, really.
I think one bit of advice is just contact people. It can be here on campus. If that doesn't work, you can try UMD. So look at or UWS. Look at the faculty lists for the biology department or whatever department that you feel most closely aligned with, and then just start contacting them. A phone call, an email. You might not get a response at all. You might get negative responses, but it just takes one that can lead you down a path that you never anticipated. That's how I ended up with my lab technician job. I just emailed someone out of the blue, cold, turned into an interview, and I got a job. So you never know. That's how
I got my. I had a break between college and graduate school and really didn't know what I wanted to do and wanted to get some more experience. And I sent out resume and cover letter to every psychologist that I could find in Duluth, because I went to school out east, but we moved here, sent one out, and ended up at Mustafa Labsi's lab over at UMD. He called, he was like, well, never seen this come through before, so let's chat. And it worked, and it was great, and we're still colleagues today, which is really fun. So don't be shy, don't be afraid. I mean, the worst you hear is, no, we don't have anything right now, but just reach out, and what you need is experience. So if you're interested in one area and you can't match in that one area, don't be afraid to get some experience in a related area, because you can still get back to what you want to do in graduate school. You need experience to get there.
Yeah. If you contact faculty, if you're interested in their research, if you read about it online and you know a little bit about what they're doing, you can get them started talking about that, because that's their most beloved thing. And they will be psyched to talk about it. They will be so excited that you cared, and that's how you get your foot in the door.
So as Dr. Mackey has said, the best thing to do to get your foot in the door with a faculty member is to just approach them and ask about their research. So our brave scholar Serena volunteered a question at this time, and she asked our faculty mentor panel about timelines related to research and how to know when a project is due, or if a project timeline is indefinite.
That's funny. I would bet that there is a personality variable that's relevant in the answer to that question. I bet there are some individual differences in how people approach that, depending on project and funding, et cetera. But go ahead.
No, I wasn't ready yet.
Oh.
Dan's ready.
For me, it depends whether the research is happening in the classroom or if it's my more intensive student mentored based research sorts of questions. If it's a directed research question, we don't put any sort of time frame on that, because that can make people rush. It can make people cut corners. So I don't necessarily put time frames on when the experiment begins and ends. We certainly will set rough timelines of optimistically where do we want to be at certain times, but no hard deadlines. With the phage course, the actual structured course that students enroll in, that is a little bit different, where we do have deadlines for submitting certain pieces of information, and that always adds a little bit of stress, and it's not ideal, and we always seem to be just kind of rushing as these deadlines approach, but it's necessary to stay part of the program. So that's, I guess, my response to that question.
I always have timelines. That's my concern. But I do for everything. That's how I operate. I'm a little bit type A in that way, and I'm time pressured. And we just worked on a search with me. Does that make sense now?
Yes, it does. But it was a very well-run machine.
Why, thank you. Why, thank you. So, yeah, nobody dies if it doesn't get done in that time frame. But I feel like it's just easy to let things slip away if you don't keep
that is a very good point.
Mm-hmm.
Yeah.
I feel like there are a lot of ways to answer the question. Like if I was still working on ribosome assembly, people have been working on it for decades and decades, and so that could go on forever. It's just a basic scientific process of figuring out what the basic biology is of that organism, and so there's really no timeline for that. But if you are on that project and you have funding from the NIH or the American Cancer Society or the National Science Foundation, then you have to produce in order to keep your funding. And so there are those kind of external pressures, and Dan was talking about the phage external pressures. I also feel like my collaborative group, they can be an external pressure because they do the work every day, and I teach and run a department and run the lab, and so you can imagine who gets more work done, right, on the project. And so they will be like, hey, we need to have a meeting, like, what's going on? And, oh, dang, yeah, I'm behind. And so that keeps me pushing. It also can be pretty stressful as far as like, okay, it's advising week, I can't really meet your deadline right now. So as far as like self-imposed deadlines, I feel like I always have certain goals. Like I want to bring a student who has worked with me for 2 years, I want to bring them to a national meeting so they can present the data. So then our goal is just that they have a reasonable chunk of data that they can make a good poster, and we can be proud of it, and they can go have that experience, because the science communication is such a huge part of it. So I tend to have goals like that, which are a little bit more educationally based, whereas my folks at the EPA would be they're very product based, and so their product is a manuscript. They need to write papers in order to keep the project rolling. And so you have a lot of different goals and a lot of different challenges. I think that potentially put some pressure on the science and the pace of it. But in general, if you're doing basic research, it's just going on forever. If you go down one path and nothing seems to work, you jump paths and then you go to something else. But other than that, you could stay indefinitely on a research project. So it's variable.
Great question.
Yeah. Another question?
Are you all hitting that point in this semester where you just feel so busy?
Yeah. Us too.
I'll add just one more thing. I've thanked her before, but Jen is the one that keyed me in on the CPHAS program. So it's kind of one of those sorts of things where you don't really anticipate where everything's going to go. And if it wasn't for Jen telling me about it, it wouldn't have been there. Yeah.
And you know why I knew about it? Because I went to UMD and I gave a talk one time because I was kind of fishing around maybe for a job. I thought I might do a postdoc there. And when I gave the seminar, I met Lou Barker, and she's a phenomenal educator, and eventually she left UMD, got a job at Howard Hughes Medical Institute, and was in charge of the CPHAS program. So the whole nationwide program that Dan's working in, she basically sent me an ad that said, you know what, this is my job now, this is what I'm doing, I think you would be great, do you want to apply for it? And at the time in my career, I made the mistake of asking questions instead of just saying, yes, I want to apply for it, I'm going to do it. And I asked my chair at the time, and he said, no, not for the chemistry department. And so I pitched it to biology, like, here, you should do this. Can you do this? This would be so cool, and I'm going to be super jealous. And it worked out great. So yeah, just keeping those connections going, that's actually just a story of like connecting socially with people. I mean, I know I connected professionally with Lou, but we are friends now, and so keeping those contacts, because they'll kind of keep their eyes out for you in terms of what fits your institution. In my case, like, you're at a little small college, this would work for you, because resources can be challenging here. So yeah, I was really happy you could do that.
And for a while, it looked like we were both going to be teaching it together, but then
Until I asked permission.
Don't
Don't ask permission, ask forgiveness. So you do what you want, and then when you get in trouble, just say, I'm really sorry, I didn't know any better.
This seems a little bit contradictory to your ask a lot of questions. Ask all questions.
Not
all
of
the time.
authority. Look out for yourself. I know, you have to strike that balance.
So that brings our time here today to a close. Thanks to the U.S. Department of Education for funding the Trio McNair Scholars Program and allowing us to do this type of work. Fun fact from Dr. Ronald E. McNair's life, our namesake, talking about research. Do you know the title of Dr. McNair's dissertation? Energy absorption and vibrational heating in molecules following intense laser excitation. Dr. McNair earned his PhD from MIT. And with that, everyone, have yourself a wonderful day.